Cells of the immune system, granulocytes. So these granulocytes are mainly associated with innate immune system, and let us see why. So let us look at the learning outcomes of this session. Granulocytes are classified into three main type of cells: neutrophils, eosinophils, and basophils, with different cytological and cytoplasmic staining characteristics. In fact, their names have been given based on the cytoplasmic staining characteristics. There are within the granulocytes present granules, and again that is why the name has come up, that contain enzymes and chemicals that help in an immune response. Neutrophils represent a very high percentage of granulocytes that are circulating and take part in inflammatory reactions. Eosinophils are motile phagocytic cells that can migrate from the blood into tissue space. And basophils are non-phagocytic granulocytes, but they release pharmacologically active substances, thereby leading to an immune response. Now, neutrophils are also called as polymorphs or polymorph nuclear leukocytes. Now, when you look at a neutrophil, one of the distinct things that is there is that the nucleus is multi-lobed. And the second thing that is very prominent in neutrophils is the presence of different granules, okay? And of the different granules, some are primary granules and some are secondary granules. Now, what would also be observed in the neutrophils is formation of what is called as phagosomes, because they are phagocytic in nature. So the pseudopodia help the neutrophils to engulf the antigenic components or foreign particles, engulf it and form phagolysosomes, thereby destroying or eliminating the antigens. Now, neutrophils, like almost all WBCs, are developed from hematopoietic stem cells. They circulate in the peripheral blood for 7 to 10 hours, and then they migrate into the tissues. Now, the migration into tissues, of course, has to do with a phenomenon that is called as chemotaxis. So, there has to be certain chemicals synthesized by other cells that help the neutrophils move towards the tissues. Now, during infections, there is a transient increase in the number of neutrophils, and this aspect is used in the medical fraternity to find out whether a person has an infection or not. So, it has a diagnostic relevance. 50 to 70% of WBCs that are circulating are in fact neutrophils. They are they are associated with a phenomenon which is called as leukocyte extravasation. What does this word mean? It means that these leukocytes, which they are talking about in terms of neutrophils, are able to wade through or pass through the blood vessel lining, tight blood vessel lining, and get into the tissues. So from the circulation from blood vessels, they can get into tissues and they squeeze through the walls of the blood vessels. So that is what is called as leukocyte extravasation. Now, for leukocyte extravasation, there are three main processes that happen because of which there is extravasation. So you have the neutrophils, slowly, gradually, because of integrins bind to molecules that are present in the wall of the blood vessels. For example, integrins can bind to selectins, so you have E selectins and P selectins, and having tightly bound to these through also what is called as ICAM, that is adhesion molecules. And these adhesion molecules, the tight binding of these adhesion molecules with the neutrophil enables a process that is called as diapedesis. So diapedesis is when the neutrophil is able to squeeze through between the cells of the blood vessel, then that is what is called as the diabetes, and that is associated with the interaction of the ICAMs and the PCAMs with the E-selectin and the P-selectin. So together as a molecular mechanism, diabetes leads to migration of the neutrophil into the tissue space. Now you must understand that the leukocytes will move towards a bacteria because or move towards the tissue damage area because of the release of chemokines or the release of cytokines by other immune cells like macrophages, which are also part of the connective tissue or the endothelial linings in the body. And so the phenomenon of leukocyte extravasation is being influenced by chemokines and cytokines released by other cells, neighboring cells. And this is what is called as the phenomenon of chemotaxis. Now, once these neutrophils are present in the tissue and they encounter the microbes or the antigens, they would, through pseudopodia, engulf it. And once they engulf it, they form what is called as phagosomes. And those phagosomes then will fuse with lysosome to form phagolysosome and then break down and eliminate the antigen. So this is how neutrophils actually play a very important role in innate immunity. And as you can see, this is innate immunity because it kind of also forms or is part of what is called as the first line of defense itself. Now neutrophils, therefore, uh, can we said that it through chemotaxis it is able to function. So neutrophils can be attracted by cytokines that are secreted by T helper cells. They can interact with complement system, which are molecules of the immune system that help in elimination of antigens or foreign molecules. They are associated with components of blood clotting system, and they are also attracted to cytokines secreted by macrophages. So all of these basically help in activating or leading the neutrophils to escape from the blood vessel and enter the tissue system. So this phenomenon, because of all of this, the neutrophils moving towards the tissue is called as chemotaxis. Within the neutrophils, there are lytic enzymes and bactericidal substances that are present within the granules. The primary granules will contain hydrolytic enzymes, peroxidase, lysozymes, and all of these are involved in the phagocytosis of the neutrophils. Secondary granules contain collagenase, lactoferrin, etc. Now, in all, what has been observed is that neutrophils can kill or their antimicrobial or antimicrocidal activity is thanks to two pathways. One is the oxygen dependent killing, and the second one is oxygen independent killing. So, the oxygen dependent killing is because of the formation of reactive oxygen species, and these reactive oxygen species being free radicals, these free radicals are able to attack various different biomolecules, denature them, and in the process kill the bacteria or affect any foreign particle. In the oxygen independent killing, there are many molecules which are able to kill the bacteria through, say, for example, production of high level of defensins. And these defensins will make the wall of the bacteria porous, and thereby it would affect the osmotic balance, create what is called as osmotic imbalance in the bacteria, and thereby the bacteria will eventually get killed. Eosinophils are again similar to the neutrophils. They have a multi-lobed nucleus as well. But what is very prominent and different from the neutrophils is that it has granules which actually can take up an acidic stain like eosin. That's the reason why they are called as eosinophils. In neutrophils, the granules were not stained by either acidic dyes or basic dyes, so they were called as neutrophils. In this, all these granules contain constituents that are able to take up the acidic stain. They comprise 1 to 5% of peripheral blood cells. They help in inflammation. They are found to be having a defense against parasites, and they are able to, because they release collagenase, etc., they are able to carry out extracellular digestion of parasites. And on its surface are present Fc receptors, and these Fc receptors can bind to antibodies especially already bound to antigens. So the antigen antibody complex can bind to these Fc receptors, and when they bind to the Fc receptors, the eosinophils become active, and on activating the eosinophils, they are able to carry out phagocytosis or release inflammatory soluble factors that can kill finally the antigens. So therefore, one is very clear that eosinophils are both phagocytic as well as motile. In case of basophils, again, what one can see is that you have the nucleus, which is lobed, and plus there are several granules, and many of these granules are able to take up basic stains. So they are called as basophils. So they represent only about 1% of WBCs. They are circulating, but they are not phagocytic in function. On the surface of these basophils are again present Fc receptors, and these Fc receptors are especially for IgE. So when IgE binds to the Fc receptors, and in turn the IgE binds to an allergen or an antigen, there is cross-linking of the two IgEs, which in turn, that is bound to Fc receptors, get activated, and you have a signal transduction happening within, because of which you have a release of certain pharmacologically active compounds. Now, the granules contain chemicals like heparin and histamine, and these are involved in several functions such as vasodilation, then constriction, etc., and thereby smooth muscle constriction, etc., and therefore they lead to what is called as allergic reactions. So you have very clearly allergic response that is associated with basophils. So let us make the conclusions. Granulocytes contain specific granules that stain distinctively, and hence based on that are divided into neutrophils, eosinophils and basophils. Neutrophils exhibit leukocytosis, which is medically important. So leukocytosis basically means that the percentage of neutrophils increase when there is an infection in the body. And as mentioned earlier, they are relevant diagnostically. They are active phagocytes with more vigor in ingesting foreign antigens. Eosinophils are found to be associated with immunity against parasitic infections. They contain Fc receptors enabling targeting the antibody bound antigens. Basophils are also circulating, but they are non-phagocytic, and they have granules which can, on activation, release chemicals that lead to an allergic response. So the neutrophils, eosinophils and basophils have characteristic granules that give each one of them specific functions with the object of eliminating foreign antigens. Thank you.